Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa. 안녕하세요, and hello all. Welcome and thank you, SynBYSS people, for joining us, the 165th seminar, and I call it on Imperial College London Day. Tomorrow is the Independence Day of the United States. Independence Day is not only for the United States, but also for many other nations, including India, Korea, and other ones that used to be a colony. Being independent is generally good not only for nations, but also from the human development perspectives. For example, I mean becoming an independent adult. Research, especially academic one and basic science, should be independent of any influence, including geopolitical factors or power. I hope researchers will become independent again. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Rodrigo Ledesma-Amaro. He is a professor or reader at Imperial College London and also the director of the Bezos Center for Sustainable Protein and the Microbial Food Hub. Notably, he was a rising star speaker of this seminar series on April 27, 2023, and now became a pioneer speaker only in 2 years. He's now leading multiple big centers to make people independent of traditional foods who cause multiple issues. Multiple issues, including climate changes. Rodrigo, thanks so much for your amazing contribution to scientific society and eventually solving food shortage issues. The virtual podium is all yours now, and thank you again.
Thank you very much, and thank you for the nice introduction and continuing this series of seminars that are super useful. And yeah, it's my pleasure to be here, as you say, now on the other side. And I'm going to do a very brief talk before passing the word to Heidi, who is the real reason why we are here. So let me share my screen very briefly. You should be able to see it now. And I'm going to talk about synthetic microbial communities. So as already introduced, I'm the director of the Bezos Center for Sustainable Protein. It's a center with over 100 researchers, 40 different research groups, trying to make the future of food, try to think how we can make food that is more sustainable, healthier, affordable, and secure. But today I'm going to talk about my research group in particular, which is based at Imperial College London. And we work with a variety of different microorganisms, but mostly with yeast, a little bit of bacteria, and filamentous fungus as well. And we like to convert a variety of feedstocks, especially byproduct or waste products, and convert it into products of interest. So we've been working a variety of different product with different application and with a strong focus on food and food ingredients, as we've been discussing already with this Bezos Center focus as well. And my group works in developing synthetic biology tools, mainly for engineered metabolism, in maximizing the production of some of these products of interest for different applications, and also in these new processes or synthetic microbial communities that I'm going to tell you today a little bit about. So microbial communities are great. We find them everywhere in nature. This is how evolution has decided that it's the better way to use resources in an environment. They're complex and change constantly. But in biotechnology, we have usually trying to just take one single cell and try to to engineer itself to be able to do everything perfectly by itself, convert that feedstock into the product. We often find challenges, although we've progressed a lot and made a lot of products at scale. We think that maybe bringing some of these elements of nature and microbial communities can help us build more robust and optimized communities. But for that, we need to bring this complexity of microbial interactions to engineering biology. So we need to build new relationships within cells and new systems that allow us to build those robust communities. And one of these mechanisms is cross-feeding, which is the exchange of essential nutrients between cells. And this is key because it allows us to keep the multiple cells in the environment instead of one taking over the other. So in this example of a paper from my group, we screen a large library of knockouts to select those that can cross-feed metabolites between them, allowing them to grow only if they are together. In another example of microbial communities is those that allow us to control the population ratio. And this is important in optimizing purposes because we want to have the right combination of the 2 or 3 populations to maximize those metabolic fluxes from substrate to product. And we've done multiple activities in this regard. In this particular paper, we developed 4 different ways to control population ratio and optimize production with them.
Rodrigo?
We started, sorry?
I'm so sorry. I think I can only see your title slide. Have you advanced some slides?
Yeah, I'm advising the slides, and that's pretty
Yeah,
weird.
only see the first slide, but everything understandable. So for
Okay,
fun.
great.
It was very clear, so I wasn't.
Yeah, so I'm glad it's clear. We don't need slide at all. I know what will happen. I was sharing, it's not sharing the whole screen. What I'm gonna be doing now. So you see it now. Yeah, perfect. Yeah, so we're talking about controlling communities. Another interesting thing of communities, you can build communities not only with the model organisms, such as Saccharomyces cerevisiae, but also with other organisms. So we explored Jarobia lipolytica, we created communities with that, we were able to control population ratios as well. And then another thing we are interested in doing with communities is division of labor. So we know in nature we have different organisms specialized in different things, and this can be great for bioproduction because you can reduce burden. So we first build communities also between different organisms, so we can combine different type of yeast, for example, and then do the division of labor, where one organism can produce the intermediate and the second organism can make the product. And we found that in some cases, like this example of 3HP, we were able to produce 60 times more products with the co-culture than with the monoculture. Another advantage of communities is we can expand the utilization of complex feedstock, or in this case, simple feedstock but difficult to utilize, like CO2. So we created communities of cyanobacteria that can convert CO2 into sucrose in this case, and we engineer an heterotroph to use this sucrose to produce a molecule of interest for the food industry, beta-caryophyllene. And then we put them together, and you can convert directly CO2 into a complex molecule with an heterotroph. And the final example of things we can do with communities from my side is the upcycling of byproducts. So in typical bioprocess, you have a cell that converts substrate into a product, but cells usually like to produce other byproducts that are taking away some of the carbon away from the product that we want. So we develop a community where the second cell cannot use the substrate and has been evolved and engineered to use the byproduct as the main carbon source to make more of the product. So in the end, we are channeling this byproduct into product again. And this was done by combining metabolic engineering and evolution. And by adding this second cell, we duplicated the biomass that was generated, but also the production of the product of interest, in this case, beta-carotene. And with that, I would like to thank all the members of my groups, all the funders, but also use this brief introduction to link with another of these features of microbial communities that we're hopefully going to hear more about after my talk. Thank you for your attention.
Thank you so much. This is such a wonderful talk. The one thing I wanted to say, the best talk I listened to was in the conference was because of technical issue, the one speaker cannot show the entire slide, but she pretend showing the slide. And that's the most memorable presentation I ever listened to. And then you also did great job, because I understand everything without the slide. Also, I also want to say the food is very important issue, even though some people in developed country would not understand the problem, because we have too much food or too much food waste. But I believe that is a very important problem globally, because I will also say food inequality. Some country even do not have enough food. So I believe the microbial food generation is very important solution, and I hope, I mean, the center will succeed. In that way, we could feed more people globally. And so thank you so much for your effort. It's team effort, of course, but you are leading very important, team effort together. Thank you so much. All right. Now the main speaker of today with a longer introduction, Dr. Heidi Klumpe is a Damon Runyon postdoctoral fellow in Mo Kali and Mary Donald's group at Boston University. That fellowship is very competitive and this huge honor, I believe. She holds a PhD in chemical engineering from Caltech, where she worked in Michael Alloy's group. As you know, Michael Alloy is a father of synthetic biology and also gave a wonderful talk for this seminar series. She is interested in understanding the design principles of multicellularity that would allow us to construct communities, tissues, and organisms from the ground up. And also, like now I heard, she is very interested in organoids as well. For her PhD, she studied combinatorial signaling of a key developmental signaling pathway, BMP, showing how cells integrate combination of highly similar protein into distinct conductive responses. For her postdoc, she has been studying the effect of cell-cell adhesion on cell fitness and function with engineered yeast aggregates, which she will present, I believe, in this week's seminar. And also I heard, I mean, she's gonna be join one European institute in Netherlands soon. I want to say congrats. This is also very exciting journey for her, I guess. Heidi, thanks so much for your time today. Please take it away. It's all yours. Thank you.
Yeah, thank you, Taesuk. I really appreciate the kind introduction and the chance to talk about what I've been working on in my postdoc. And also, it's really nice to be here with Rodrigo, who has kindly shared strains with us, and we've talked about the project together because we're sort of interested at the intersection of engineering yeast communities and thinking about metabolism and aggregation. So I'll share my slides, and please let me know if I have any technical difficulties. But I think, okay, we can see title slide. Okay, great. My name is Heidi Klumpe, and I'll be talking about my work at Boston University, where I've worked with Mo Khalil and Mary Dunlop. And partially because I did my PhD in development, I was interested in kind of thinking about what were some like basic principles for understanding how to construct something multicellular. And I really kind of went maybe very, very far at the other end of the spectrum, thinking about microbes and this really basic feature of multicellularity, cell aggregation. And one reason I thought it was sort of cool is that even though cell aggregation or cells being close to each other is a key feature of multicellularity, which we usually would think of as something that is like more useful for cells, kind of like Rodrigo mentioned, we want to get kind of closer to nature and more of its complexity. But cell aggregation induces both costs and benefits. So it's unclear sort of what effects it will have on cell state. And so I think cell aggregation is interesting because it fundamentally changes cell state in two ways. So one of them is separation from the bulk media. If molecules can't penetrate to the inside of the aggregate, which is beneficial if you want to be protected from something in the environment, but can be costly because then you have reduced access to whatever nutrients are in the bulk media. Another thing that's very different in an aggregate relative to individually suspended cells are proximity to other members of your population, which just sort of gets to what Rodrigo was talking about. This can be excellent if you want to do metabolite sharing or have some division of labor, but could be problematic if you're competing for a finite resource pool and you're quite close to each other. And so I think something that underscores this reality that even this may be a simple change to bring cells close together can have unexpected effects is if we look at experimental evolution, which really shows how aggregate fitness can be context dependent. So something cool is that even in the timescale of a laboratory experiment, you can watch aggregation evolve, and usually it's in the presence of some kind of toxin or predator. So the example I'm highlighting here is Candida auris, which is a fungal pathogen, and it evolved over the course of infecting a mouse to aggregate and stick together. But there's other examples in bacteria with E. coli grown in high salt will also start to aggregate as a byproduct. And this green algae, Chlamydomonas, if co-cultured with a filter feeding predator, will also start to aggregate. So these seem to suggest that aggregation provides some benefit if you need to be protected from your environment. But by contrast, aggregation can also be lost, in particular if you're continuously culturing in rich media or some context where aggregation provides no apparent benefit. So this is an example of a different type of yeast, Saccharomyces cerevisiae, which is the one most people are familiar with, which starts as these evolved snowflakes, which don't finish the final step of cell division. So basically, they all stay stuck together. And over time, they will also revert back to being individual cells. And there's also examples like Pseudomonas fluorescens doing a similar reversion from being aggregating to non-aggregating when they're grown in unstirred media. And so what I think this suggests is that we need to better understand how the costs and benefits of aggregation arise in order that we can better study different types of cell aggregates, but maybe also engineer them. I mean, even looking at the prior examples from experimental evolution, it suggests that there are maybe rules about media content in terms of both toxins and predators, but maybe also media richness. We suspect also that because some of these things are related to diffusion, there might be characteristic length scales where they occur. So aggregate size maybe also matters for determining whether or not that aggregate is overall beneficial. And lastly, aggregation mechanism probably we can imagine would matter in different ways, both for doing things like tuning the size of the aggregate, but also how costly is it to produce that molecule. Biofilms often stick together by these like long, sticky polysaccharides, which maybe are more costly to produce relative to just some sticky proteins. But again, I think it's sort of interesting to think about these things because useful cell aggregates need not be an entire organism, but we might be interested in engineering strains for bioremediation that are going to be grown in toxic settings, and maybe aggregation could improve their survival. We're also interesting, like what Rodrigo was talking about, is engineering cross-feeding, and if that can be done more effectively in a group of cells stuck together, that could also, maybe boost manufacturing. And then also kind of thinking in the human health domain, thinking about like drugs entering tumors or how effectively cancer cells get sugar is also really relevant, and which is what I sort of wrote my Damon Runyon proposal about, was thinking about how can we think about sugar and oxygen and yeast aggregates as a model for what happens in tumors. And then also it's, I think also well documented that in organoids, there's also problems with oxygen delivery. And that's also what I've decided to work on for my second postdoc. But this is just to give a sense of how I think these maybe kind of maybe more microbial or like simpler examples, I think have broad implications for all the different types of cell aggregates that are out there. And so for this project, I was really interested in something where we could have a bottom-up approach, because I think the experimental evolution is really exciting. But when we use synthetic biology tools to make really precise perturbations and control the different strains that we build, I think we can kind of learn more, get more quantitative insight and more strong evidence of causation. And so the idea here was to look at one particular aggregation mechanism, cell-cell adhesion, in part, I think, because tissues are held together by adhesion proteins, but also slime molds and yeast, and then use this sort of minimal model of aggregation to really ask, okay, what are the effects of cells sticking together and forming aggregates. So one thing we can ask is compare, our aggregates with individual cells and ask, are there actually effects on growth rate, which would be a high-level readout of effects on fitness? Do we see any evidence of protection? Is aggregation sufficient, protect you from something toxic in the environment? And lastly, can we see any evidence of collaborative metabolism, that cells are able to extract more energy from the environment or just do that more efficiently as a result of being aggregated? And as I've sort of teased in the title, yeast flocculation ended up providing a great model for studying this and kind of getting at these questions. So one is a biological reason, which is that yeast do this context-dependent aggregation that I mentioned before, and that they flocculate proportional to the amount of ethanol in the bulk media, as is shown by this nice figure from this paper from 2008. And this kind of matters also because we know that ethanol is toxic to yeast. So this seems in line with the experimental evolution examples. And it's also, on the technical side, a good reason to work with it is that it's fairly easy to engineer. You can just engineer expression of this one of the flow proteins, and you'll get an aggregate. Okay, so this is why we chose to work with yeast and these flow proteins. Prior work had focused a lot on flow1, but there are other flocculation proteins in yeast. So we looked at expression of all of them. They vary in composition of the stalk, the bit of protein that extends the sticky bit away from the cell wall. So again, you should think of these as like proteins embedded in the cell wall on the yeast. But the main difference, I think, is between the adhesion domains. So 3 of them have this PA14 non-specific domain that just sticks to the yeast cell wall. So we'll stick to any yeast, whereas flow11 is more specific, and it needs to stick to another flow11 domain presented on the opposite cell. But one sort of surprising thing, as it says in the title, is that even fairly similar flocculation proteins, when we express them, didn't necessarily produce aggregates. So we were somewhat surprised that flow10 didn't seem to work. Which I think kind of gets to results that when we're back in the literature, I guess it's some people have reported for different flow proteins that the composition of the stalk actually matters, which is sort of cool if you're thinking about engineering flocculation proteins, which is actually something Rodrigo's group has also done. But for us, we thought, okay, we'll just focus on looking at flow1 for now, because it definitely forms really, really large aggregates. And just a quick reminder that actually for all of the microscopy, even though the aggregates are 3D and they form in stirred media, to get the images, we actually carefully flatten them into a 2D monolayer, and that's what we image. So you should when we want to quantify aggregate sizes, for example, we look at the 2D area, but you could kind of think about that as a sphere and it's 3D area. But the flow1 aggregates were large enough, you didn't need a microscope to see them. But we were interested in asking, can we control the size of the aggregates? Because again, we had intuition that a lot of interesting aggregate properties scale with size. So we used something cool. If you're working in yeast, this yeast toolkit, we have promoters of different strengths. And the idea being that if you have less flow1 on the cell surface, that cell is overall less sticky, so you would get smaller aggregates. And indeed, we found that as we tuned down the expression of flow1, aggregates got smaller. There's also this co-transcribed RFP. So we could also check by flow and a little bit by microscopy how much flow1 is there. Another important thing we found is that the culture density also matters. So these are quantifications from microscopy images. It's kind of a histogram of sizes. The black histogram is the non-aggregating control. So you can see that it gets bigger as you increase the concentration of flow1. This medium OD is quantifying the images that you see here. But you can, of course, make the culture more dense, and then the aggregates get even bigger. And if you have a less dense culture, even a little bit of expression of flow1 isn't sufficient to produce aggregates. But these are basically the 2 experimental handles we have to tune aggregate size. Okay, so the first thing we wanted to check was, did we see any effects on growth rates? And we had 3 different strain classes we wanted to compare. So we had different strains that didn't express flow1, variants of our base strain. We have the strains I just showed, which express flow1 at different levels, thinking maybe you have to express flow1 very highly before you see a fitness defect. And we also have this flow1 mutant where we've deleted the PA14 domain. This schematic isn't exactly to scale. It's basically a protein of very similar size, but we thought that was a nice control for a non-aggregating population that is expressing a similar protein. And so what we did was, over time, we would sample the aggregates, break them up with EDTA, which chelates the calcium they need to aggregate, and then look at increases in OD over time, and then we can fit these growth rates. But the punchline is basically that we didn't see any differences in growth rate, even when we expressed flow-1 very highly relative to expressing a mutant or expressing nothing at all. And the other cool thing is we did these experiments. We had wells that started at sort of low OD of 0.25 and also a higher OD of 1, which you can think about the prior slide. We know that the aggregates are much larger, but even then there wasn't much of a reduction of growth rate. Okay, so then we thought maybe we need a more stringent test of effects of flow-1 on fitness. And we thought about an experiment sort of in the style of experimental evolution, where we would just culture our engineered aggregates continuously in rich media, where again, if there is some sort of fitness defect associated with flocculation, we would expect to see it to eventually be outcompeted. And the idea here is that, there's a spontaneous mutation happening all the time. We think there are some cells that don't express flow-1 that might pop up, would we see that those eventually take over and there would be some loss of aggregation? And this was really made possible because of the Evolver, which is a platform for automated continuous culture developed in the Khalil lab. And the nice thing is that we can, because there are multiple vials that are being maintained at the same time, we could look at our aggregates of different sizes. We had replicates. And the other nice feature is that because the dilutions are automated, we can actually keep it in a narrow window of OD values, so the aggregate size is approximately the same throughout the experiment. So we basically maintain them constantly in exponential phase. And kind of like the prior experiment, we did these all in rich media, with the idea being that the penalty for aggregation is larger when you're growing in a media where the most advantageous thing to do is just import whatever you can from your environment rather than synthesizing it yourself. And so just cutting to the chase, we actually found that even if we took through our 3 different strains, so no flow-1, medium flow-1, and high flow-1 expression, which at the beginning of the experiment could still form fairly large aggregates. This is, an 100 micron scale bar. After approximately 450 generations, even these very large aggregates were still aggregating quite a lot. So this is consistent with the similarity of growth rate, that there doesn't seem to be much of a disadvantage or fitness defect associated with aggregating. And this is just further quantification to underscore that, because these pictures are just from a single vial, but we actually had multiple replicates. And indeed, if you look at the high flow-1 expression, the distribution of sizes for the fresh overnight culture is largely overlapping with what we saw at the end of the experiment. Okay, so at this point, we were a little bit surprised because we thought, we did think that, forming an aggregate with hundreds of cells in it seems like a large perturbation, but maybe it's affecting other biological processes that we just can't detect, because looking at growth rate is sort of a very high level output. And maybe there are other processes that are affected, and we could see that if we looked at gene expression. So we decided to do bulk RNA-seq, and we wanted to, of course, take our strains that don't express flow-1 and then the high flow-1 expression strains from the prior experiments. We took them from exponential culture, but we also wanted to look at higher ODs, or saturated culture. And the idea here was, we wanted to look in kind of the largest aggregates we could make, which is both by kind of maxing out flow-1 expression and also making the culture more dense. But as a control, we also wanted to include our non-flocculating strain also at high culture density, because we know that sort of as you shift from sort of lag to exponential to kind of this more saturated phase, there are huge effects on gene expression. So we really want to isolate the stuff that's specific to flow-1. So we have 4 samples and 3 replicates for each. And so we do the RNA-seq and we quantify each gene, and then we can, for each gene that we have in our transcriptome, look at expression across these 4 samples. And what we did was we actually fit a linear model of gene expression that allows us to separate what's the effect of flocculation from what's the effect of density. And you can see that for each gene, basically there are 4 classes it might fit into. So there are some genes that aren't really affected by either flocculation or density. But then there are some genes that are specifically affected by flocculation. So you can see here that this gene is downregulated when flow-1 is expressed. There's a difference for the marker style. And that's true whether or not you're in exponential phase or saturated. And then there are genes that are unaffected by flow-1, but really just vary with culture density. So just the colors are different. So this elongation factor is downregulated when the culture becomes saturated. And then there are genes that are a combination of both. And this is sort of cool that you can see it is almost a linear product of upregulation due to flow-1 and sorry, not a linear product, a linear sum of upregulation for flow-1, or sorry, downregulation for flow-1 and upregulation for density. But even though I can show you these examples, what we actually found, if you look across the whole transcriptome, is that it's very rare to find any gene that was affected by flow-1 in a statistically significant way. So, we look for a p-value adjusted for multiple hypothesis testing, and we also said we want the biological effect size to be at least a 50% change. And basically, then you get a list of 16 genes. And this is very different from what the genes we found that were affected by cell culture density, which is basically half of the transcriptome. The other point is that not only are very, very few genes affected, if we look at those genes, the effect size is very small. So like how much gene expression changed didn't seem to be a lot. So it really seems it seems unlikely that, we think that like there's a strong effect there. So this again was in agreement with what we'd seen before, very few effects on growth rate, flow-1 expression seems to be stably maintained, and very few genes are affected by the expression of flow-1. Okay. But even in the absence of some of these maybe more biological effects, we could still find physical protection because we don't need a change in gene expression for this kind of cluster of cells to be protected from something in the environment. So we then wanted to check if flow-1 expression could protect cells from toxins. And there's some evidence for this in the literature where one group expressed flow-1 and showed protection from ethanol and also this fungicide, amphotericin B. And another group was looking at a wild flocculating strain, but they also showed some protection from acetic acid. And for these experiments, just a small note that we actually shifted to a small molecule inducible system we have. This gives us kind of more, we can kind of take the same strain and get aggregates of different sizes from it. And it also produces even larger aggregates than this PTEF1 promoter that I've been using in the talk so far. So just to note that these are the largest aggregates that we can make. And we, of course, for our aggregating condition, are looking at induction of flow-1, but then we had various negative controls that were helpful, looking at induction of the flow-1 mutant, just M. scarlet alone. And we also have an uninduced strain, just to look for fitness defects associated with the induction itself. Okay. And we were somewhat surprised to find that even when we were kind of maximally inducing expression of flow-1, there didn't seem to be any improved growth rate in ethanol. So here we have 4 different colors showing these 4 different strains that I mentioned. So 1 aggregating, 3 non-aggregating. And you can see that the growth rate, or like the slope of this line, sort of decreases as I increase the concentration of ethanol. And I think this summary of the best fit growth rate shows well that again, growth rate decreases with ethanol, but it's the same for all the strains. So it doesn't really seem that flow-1 is providing any protection. Of course, it's also important to look at viability. So we also did limiting dilutions, so we could quantify the number of colony-forming units per milliliter. But we basically see the same effect, that there's some increase in viability when the cells are growing without ethanol. There's less of a viability increase as I increase ethanol. The main difference is that actually at this high ethanol concentration, we see that the cells are indeed dying. But again, there's not any protection associated with flow-1 expression. These data are kind of noisy, but what we do indeed see is that it's similar levels of growth across all strains. So flow-1 is not really providing any protection from ethanol. We then thought maybe the problem with our experiment was that the aggregates were too small. And so we just needed to make them bigger, and then we would start to see some of those diffusion limitations. And we've already maxed out flow-1 expression, so we increase the culture density. And so these data, the circles are the same data as in the previous slide, and I'm looking now at the fold change in viability. So you see that in the absence of ethanol, the cells grow a little bit, and at 15% ethanol, there's some death. But then the X's show the same experiments, but the initial cultures are at a higher density, OD5, which is, again, close to the maximum in YPD of 10. So in the absence of ethanol, they don't really grow much because they're very quickly using up all the resources that are in the media. The thing that was interesting is that these higher density cultures, when we grew them on 15% ethanol, actually died more slowly. So there did seem to be some protection at high culture density, but it wasn't because the aggregates were larger. Everything was much more protected. And then we thought, okay, the other thing that might be important for chemical protection is the diffusion rate, because in order to get some diffusion limitation, you need consumption to proceed more quickly than the diffusion. And we knew that larger toxins would diffuse more slowly. So we thought, okay, let's look for survival on this fungicide, amphotericin B. And also, we're going to try this proprietary mix of enzymes called zymolyase, which you use to basically digest the cell wall, and also will kill the cells. But what we found is even for these larger toxins, it's basically the same type of trend. So here, this is an increasing concentration of the fungicide. And as you increase it, you see that the fold change in viability goes from larger than 1 to less than 1 if the initial culture density is about 1. But if we start with a higher culture density, again, there's not much growth, but there's also not much death, which is pretty striking. But again, it's not specific to flow one. It just is because something to do with the culture being higher density. We also checked for zymolyase. This experiment is a little bit different because the YPD inhibits the rich media inhibits the activity of the enzyme. So we did it actually in buffered salt solution. And we also did it over a shorter time period just because we had problems with the flocks getting stuck to the walls. But it's a pretty similar trend that at low OD, there's some rate of death and there's slightly higher protection when you increase the culture OD. I think the main thing that's really striking here, and maybe you can see a bit here, is that the cells that seem to be most protected are actually the uninduced cells. So potentially there's some fitness cost associated with induction that may increase the vulnerability of these strains to zymolyase or just their overall survival in this salt solution. But again, no protection associated with FLO1 expression. Okay. And so we were trying to make sense of this. And one cool thing is that the papers that I mentioned at the beginning, where they reported, protection from some kind of chemical toxin, they also reported which genes they thought were important for protection. So in this case, the acetic acid case, they identified some stress kinases that if you overexpressed them, that was sufficient to protect the cells. It wasn't actually the flocculation that was important, it was these stress kinases. And when we looked in our RNA-seq data, those stress kinases are actually upregulated specifically when the culture is saturated, and actually has nothing to do with FLO1 expression. So this is sort of consistent with basically the genes they say should be upregulated when you get protection are also the genes that we would say are upregulated because we only saw protection in the saturated or high density case. Another cool thing is the paper that had looked at protection in ethanol actually did a gene array. So they had something like the full transcriptome and kind of the reported changes in gene expression for FLO1. Those actually correlate pretty well with the full changes that we found for culture density. So it's not even saying that the same genes changed, but actually the amount that each gene changed was about the same, which is pretty striking. So I think there's sort of two possible interpretations of what's going on here. And I think the main takeaway is that separating the effects of aggregation and culture density is going to be really important for actually understanding if aggregation is useful and is something we should use, for example, to boost, say, the survival of our strains in toxic media. And one possibility is that there are overlapping effects because when you bring cells together in an aggregate, you create locally high culture density. So maybe it's not so surprising that something that might be attributed to FLO1 could just also be attributed to the culture being more dense. But I also think that there's a possibility of experimental bias because aggregates are largest at high culture density. So you might be sort of working with your strains only in this regime and then kind of misidentify the effects. I think the multi-factor analysis where you separate aggregation from culture density is quite important. Okay. So we kind of we found that there aren't many effects on growth rates and there's limited protection in the flocks. And sort of the way to understand these things, something that would help us think about that would be to know how well molecules can enter the aggregates. Like, are there actually any kinds of diffusion limitations? And again, we had intuition from even papers looking at other types of yeast aggregates that it was difficult for certain molecules to get inside the aggregates. So this is a snowflake yeast that has a mitochondrial reporter, basically showing that aerobic respiration only happens on the outer edge. So it seems like oxygen doesn't diffuse to the center of this snowflake. And this is a yeast colony grown on limited glucose, so about something like I guess like 5% the amount that's like normally present in rich media. And again, only the cells on this like outer periphery are growing. And our flocks are large enough, or that they're definitely bigger than the length scales of these gradients. So we anticipated that there would be some kind of diffusion limitation as well. So we wanted to ask, how well do molecules of diverse sizes enter flocks, and maybe also just check for oxygen and glucose transport into the aggregates. So firstly, to check molecules of different sizes, it's just a happy coincidence that there are these yeast cell surface dyes that are very different molecular weights. And so much like the toxin example, we thought, okay, we can check by doing like a brief pulse of dye if there's a difference in how long it takes kind of a larger and more slowly diffusing dye to stay in the aggregate relative to a smaller and more quickly diffusing dye. And so for these experiments, in order to get this like 1 minute pulse of dyeing, we would quickly dilute with YPD 1 to 10. But then we thought maybe there's like still some amount of background dyeing that occurs while you like process the cells and get them ready for flow. So I also took some dilute dye and put them on unstained cells just to get a sense of the background, which is what these X's are. But basically you can see that the degree of staining, the percentage of cells that are stained, is the same for non-flocculating strains, and then medium and high expression of flow 1. And that's for, okay, so that's for a dye that's already 900 Daltons. We also had wheat germ agglutinin, which is 40 kilodaltons, and you see the same degree of staining across all the aggregate sizes. The only time we saw exclusion was actually for concanavalin A, which is 100 kilodaltons. And this was actually quite a nice result because it actually scales with the size of the aggregate. So there's pretty good staining of individual cells, slightly less staining for medium aggregates, and even less staining for the large aggregates. And what's also nice about this is this maybe agrees with our prior result about protection, because this dye is about the size of the fungicide, amphotericin B, and this dye is about the size of zymolyase. So this is consistent with the fact that we didn't see much protection because, again, molecules of the size are readily entering the aggregates. Okay. And just quickly, we also got from a collaborator at Oxford University, Miquel Livia Puerta, this dual fluorescence oxygen reporter, which is basically you can look at the ratio of GFP and RFP as a readout of intracellular oxygen. And I also worked with Daniel Hart, who's a grad student in the Khalil Lab and has an evolver where you can control oxygen levels. And we confirmed that as we removed oxygen, indeed, this GFP to RFP ratio shifted and increased. And then what we wanted to do is then check, okay, for different culture densities. So really ranging from very, very small or non-existent aggregates to large aggregates, do we see any difference in the distribution of intracellular oxygen? Like, is there a population that shows up where it looks like they're starved of oxygen? But that wasn't the case, even looking in glucose or non-fermentable glycerol. So this is sort of surprising given how big these things are, but definitely consistent again with, no difference in growth rate and very few changes in gene expression. Okay, this is sort of the final part of the talk, but this is actually kind of one of the first clear differences between aggregating and non-aggregating cells that we found, which was when we tried to grow them on limiting glucose. And here the intuition was, based on the yeast colony paper, was that as we reduced glucose, how deeply, I guess, it can penetrate into the aggregate will go down. So the penalty associated with aggregation should be larger when there's very little glucose. But we actually found was the opposite. So I apologize because the colors are different, but it's we've gone back to the inducible strains, and I'm inducing expression of flow 1 for aggregates, or just M scarlet for a non-aggregating control. And this is their growth over time in 0.25 millimolar, so very, very little, possibly the minimum amount of glucose required for cell growth, up to 16 millimolar, which is also, I get about 20% of what's in regular yeast media. But what you can see here is that the flocculating strains continue to grow, whereas the M scarlet strains just are like stuck in lag phase, are no longer growing. And maybe in the 16 millimolar case, definitely growing a little bit, but much more slowly. Something else sort of cool would be, okay, it seemed like this effect was larger if sugar, like the benefit was greater if there was even less sugar. And so we checked kind of across a larger parameter screen, looking across initial ODs and different glucose concentrations, and indeed found that sort of the benefit of being in an aggregate increases, or is the largest when there's very little sugar or when there's very few cells to begin with. We also thought maybe this effect was specific to glucose. So we checked for kind of similar low initial ODs, so a very sparse population, and low percentages of different carbon sources, other sugars, even these like non-fermentable carbon sources like ethanol and glycerol. But in all these cases, the aggregates grew better than the non-aggregating cells, which was quite cool because it suggests that maybe whatever mechanism this is might be general to, I guess, different metabolism. And so unfortunately, we don't have, this is a newer finding, and we don't have like a full explanation of what's going on. But one thing I think is cool is that it's very consistent with like another model that people use to study the benefits of multicellularity, which is how well they can capture extracellular metabolites. There's this example of growth on sucrose, which is a disaccharide that becomes glucose and fructose, and how well cells can grow on sucrose. Basically, they do it best if they have a high concentration of sucrose, which helps them capture the glucose and fructose, or there are more cells. So there's more cells producing glucose and fructose, and they're a little bit closer to each other. So this is sort of, I think, my mental model and also the field's mental model of like extracellular metabolism. If you want to improve capture of some like self-made metabolite, you can aggregate, so stick to another producer of that metabolite. You can increase the precursor concentration in the bulk media, so cells can just make more of this because they have more of the reactant that they need. Or you can increase the cell density just by having more point sources. Again, the bulk media is higher. So I guess we're not really sure what is the thing that the cells might be sharing within the aggregate, if it's like a secondary metabolite they can capture energy from, or some kind of cell signaling that's like changing the regulation of lag phase. But I think we're sort of curious to explore this a bit more. But it also suggested to us that flocculation might improve growth on sucrose, which has been shown for snowflake yeast as well. And that's basically what we're seeing here is usually when cells don't aggregate, they grow okay on a mixture of glucose and fructose, which is sort of the positive control for the most growth you can see. But then they grow less well on sucrose. And it's a little bit probabilistic because we have so few cells to start with. Versus when the cells express FLO1, not only do they grow better on glucose, which we knew already, but their growth on sucrose almost exactly matches the growth on glucose and fructose. So they seem to be much better at capturing it. And this is just showing that similar scaling that I'd mentioned before, that the benefits associated with flocculation just increase kind of in more starved conditions. So when sugar concentration is low or when the initial OD is low. Okay. And then I'll just end with like one more result, which I think is sort of cool and unexpected, which is if we think that the primary mechanism for better growth on limiting carbon has something to do with the cells being close to each other. And we also know that FLO1 binding is nonspecific, which means that it will stick to other cells. What happens if we mix these things together? Is the benefit also nonspecific? So these are microscopy images of mixtures of aggregating and non-aggregating, or FLO1 and not FLO1 expressing cells. And you can see that basically cells that don't express FLO1 are readily incorporated into the aggregate in sort of a random fashion. And so it seems that they could also benefit from whatever molecule that is being produced and shared among FLO1 expressing cells if they're also there in the aggregate. So we made mixtures of our inducible strains, so the one expressing FLO1 or M scarlet, and then induced them as a mixture and grew them in 1 millimolar glucose, and then used flow cytometry to track the proportion over time. And we did, so I guess here's the 9:1 case. So it's mostly FLO1 expressing, not much change over time. The main difference is that kind of on like day 3, we saw that the induction circuit seemed to be broken. Again, we don't know if maybe there's some small cost associated with inducing it, but this just shows up at later times. But what was cool is then in the 50-50 and even the only 10%, sort of aggregating population, the FLO1 cells pretty drastically and quickly outcompeted the non-FLO1 expressing cells. So again, I just thought this was very cool because FLO1 binding is non-specific, yet for whatever reason, maybe because of geometry or whatever it is, the metabolite that's being shared, it gets a specific benefit and is able to outcompete the non-aggregating cells. Okay, so just to summarize, cell aggregation, I think seems like this big phenotypic change, and it's something that's really essential for engineering multicellularity, we found that it had surprisingly limited effects on fitness, that there was basically the same growth rate as individual cells. Aggregation was maintained over 400 generations and had expressing FLO1 had pretty limited effects on gene expression. And kind of these two differences that we thought would matter, like separation from the bulk and proximity to other cells, the main thing that seems to matter is this possibility for nutrient sharing, and that actually there's not much separation from the bulk. So again, we found that there was rapid staining, even with very large dyes. There's limited protection, even from protein-sized toxins, and there's not many effects on intracellular oxygen. But we did see that there are these benefits associated with kind of improved capture of extracellular metabolites, but they seem specific to something when the concentrations of these things are low. And so maybe there's some interesting modeling work to be done there to figure out kind of that scaling. But yeah, we saw better growth on limited glucose and also sucrose. And then just as a final thing, I think if we had to bring this back to something that's a design principle, again, I was really hopeful that we could use yeast to learn some kind of universal principle that might also help us think about tumors, for example. But I realized kind of over the course of this work that, there are many things about flocks that make them different from tumors, but I think they highlight maybe what are key kind of parameters to think about if you want to build an aggregate. But the flocks, we definitely have the flow of the stirred media. So convection helps mass transport. Because the cells are basically hard spheres, because the cell wall is quite rigid, you always have a large void volume of at least 20%. So it seems like liquid can get in pretty easily. And also FLO1 binding is reversible. So we know that cells or chunks of aggregate can fall off and reconnect. So that also increases their access to whatever's in the bulk environment. And I think the last thing I want to say is I think this third one might be the most important, because we can compare to the snowflake yeast that have been really well characterized by Will Ratcliffe's group at Georgia Tech. And they're basically the same in terms of void volume and being stirred. The main difference is whether or not the cells can rearrange themselves. So maybe this is a key principle that allows you to get benefits of aggregation while also reducing some of the costs. Okay, thank you all for attending the talk. And I wanted to just acknowledge the people who made this possible. So again, I work for Mary Dunlop and Mo Khalil at Boston University, and I've worked closely with Dan, Sarah, Will, Kai, and Brandon. Mikhail at Oxford gave us the dual fluorescent reporter, and these are the funding sources. Yeah, I'm happy to take any questions. Thank you.
Fantastic talk. Very interesting. I'm not sure Rodrigo's still around because he said he need to taking care of his kid. Rodrigo?
I'm listening. It was a great talk.
Do you have any question or comment?
I have too many, I think. I don't know. I first saw the question in the audience. If not, I can ask some.
Yeah, you can ask some question. I have many questions as well.
So, to which extent do you think you can use yeast as a model for cancer, right? You mentioned the importance of aggregation, like cancer and the oxygenation and nutrient dispersion and so on. Do you think yeast could tell us something about what is happening in tumors, or do you think it's better to go and straight using mammalian cells to study that system?
Yeah, I think particularly basically once we got kind of deep into thinking about I think yeast are a nice model of some of the core metabolic processes like aerobic respiration and using glucose. I think these are kind of more similar across cell types, but I think this fundamental barrier of the yeast packing as hard spheres and having this large void volume, I think means that they're not going to be a good model of diffusion limitations, which I think are probably kind of one of the key ways that aggregation affects tumors. So I think, you need, my feeling now is you need something that kind of more closely physically approximates kind of the tumor environment.
That makes sense. And just one other question, have you considered looking at single cell RNA sequencings to kind of know what's going on at different parts of the aggregate or things like that?
We haven't, but I guess I'm not optimistic that we will find something, I guess, because of the kind of limited effects on fitness. If it's anything, it's a very, very small population. And it was quite cool because the oxygen reporter is a single cell measurement and we don't see any interesting subpopulations there. So I sort of, yeah, I'm not sure we would find something, so we haven't looked at it yet. I mean, one cool thing I think has been looking at these mixtures of flow1 expressing cells and not flow1 expressing cells. And we have some fluorescent reporters there. So I think that's kind of an interesting single cell measurement because we've noticed things that like the cells will change size a little bit, which we can see by forward scatter. We've also noticed that flow1 expression seems to change a little bit, or at least like RFP, which is like co-transcribed, is also going up or going down, and trying to ask, does that change depending on whether it's like in a community or not? I think like in the cross-feeding system, it might be quite cool because if like one strain has to grow kind of more quickly than the other to maintain a balanced flux, you might see different changes in volume.
Yeah, thanks. That makes sense.
So that is all you have?
Yep.
Okay, so great. So let me ask some question because that was so interesting to me. I was also thinking about from a little bit different perspective. So the question I have is when I was in college, I basically living in a society where too many demonstrations happen to, obtain the democracy at the time. So I'm thinking somehow there should be some universal principles across different kingdoms of life, from human to animal, all the way to bacteria or the yeast. So one thing I kind of thinking about during entire your talk, uh, the question you probably didn't try that one, but who sacrifice? By that I mean who are the organism or people that is the outside of that aggregation? Is it stochastic or I just want to know your insight because you didn't have any experimental data, but is it stochastic or is it kind of some designed, genetically designed or somehow or epigenetic? I'm just curious.
Yeah, so I think in ours it's totally random,
Random. Okay.
but we also destroy the structure a little bit when we image them. So I can't say that with like too much confidence. I think in the 2008 paper from Kevin Versteppen's group that I cited a couple of times, they grew aggregates like basically the entire overnight cultures in one aggregate, so it's like a millimeter across, and they cut those in half. And I think they argued that the outside is enriched in cells that don't express flow1 because they have
Uh-huh.
kind of lower affinity.
Uh-huh.
So it's possible that because like a cool thing that happens in tissue patterning is this thing called affinity sorting, which maybe you know about, which is I think Wendell Lim's group, they have this clip where they express high and low affinity cadherins, and you get sorting of like high affinity in the center and low affinity on the outside. And so that's basically an example of that, where cells that express flow1 have high affinity for each other, and then the like non-specific interaction is lower affinity. Um, I just think that I think the stirring is maybe like mixing things up too much, so it might not be maintained for a long time.
Mhm.
And I think the affinity sorting might also rely a little bit on being able to pull on the cytoskeleton, which the yeast don't really have. So anyway, yeah, I think it might be hard to achieve that kind of patterning. Um,
Uh-huh.
another uh, like really appealing idea is like if we could get some kind of nutrient gradient inside the aggregates, you could imagine tying that to expression of something. And that would be sort of a cool way to use aggregation to induce differentiation. If you say like, hey, if in the absence of oxygen, turn on this gene. And I think there are people working on that in the snowflakes because there is a clear difference from the inside and the outside. Um, but the reason that arises, I think, is mostly because the cells can't change places with each other. Like if you're in the center, you're pretty stuck. So I sort of wonder because people talk about kind of simple multicellularity and more complex multicellularity, and
Mhm.
maybe something important is holding cells in place. Because like this the flow system is like, it feels like a pretty open, easy to use, very modular platform because there's like no effects on fitness, but you don't get any patterning. So like basically you're just stuck with like the metabolic benefits that you get. But if you want to do patterning and hold things in place, maybe you get kind of more interesting functions, but then you actually have to start dealing with the diffusion limitations. And so you need to evolve things like a vasculature or maybe, control the intercellular space so that things can get in.
Got it. That's very interesting. So the reason I'm asking that question is I want to know some governing principle across the kingdom of life, but at the same time, there should be some difference, among these, different organisms. Because, for example, demonstration, the upfront, basically some leaders, basically, upfront. In the animal kingdom, I don't remember exactly who are the outside. But there are something in outside. I mean, that's kind of evolution kind of, make that way. In that way, somehow, protect entire population. Bacteria also make the biofilm. I believe that that's similar to this yeast flocculation. So there are something, common. And because of that, I was not surprised when you're talking about glucose effect because the harsh condition to me probably trigger more aggregation, based on my kind of, the intuition. So, but very interesting. But it's very difficult experiment, I understand, because, as you can imagine, if you have too much force, I mean, the demonstration, just shattered. I mean, but it's depend on how much force, how much, toxin concentration that is important. So my guess, better experiment might be changing the toxin concentration all the way to the very low level. I mean, that may have some effect or better kind of observation in terms of observation, the aggregation, I believe. So, or looking at the death rate, probably better than looking at growth rate because I mean this is all about survivor. I mean that's my intuition, but I don't know. I mean you already done many years of experiments, you know better than me, but my intuition basically tell me very subtle condition or borderline condition may give more insight regarding aggregation than very high concentration or because of high concentration toxin that kill everybody anyway, for example. So that's sort of my general question. Uh,
Yeah. I mean, I wanted to say, I mean, we did actually, I think that's kind of what we tried to do. I'm gonna find is it this slide? Yeah. I mean, because we go for all of these, we do look, I mean,
oh, I
I
see.
always go to a concentration where death is clear, but then
Hmm.
we do go to like a
Very low.
smaller, less
Hmm.
of effect concentration. Um, so this is viability. So I don't know if that's what you mean by death rate, but we are looking at kind of at the number of viable cells at the beginning and at the end, and we're sort of spanning conditions where they grow and conditions where they die.
Hmm.
Um, but yeah, it's a hard experiment because
Yeah, it's
probably
a
both are
hard
going
experiment
on.
because you already done, you've been in the Eloise lab, there are some sweet spot you could observe some phenomena, interesting phenomena, right? But that's so sometimes very small, that area in the whatever,
Yeah, where we're at like
space.
precise point.
Yeah,
Yeah.
so I mean, this is not easy experiment to do, but I do understand. I was very interested in those kind of phenomena 10 years ago, but I realized it's too challenging, so I moved on to the more application-oriented, project later on. That's fantastic, but I wanted to be mindful. I have multiple more questions, but I will probably ask after, we close. Um, because I mean, Rodrigo should go anyway. So let me close. Okay. Thank you all for joining and staying today. We'll meet again on July 10, Thursday, the same time, the same Zoom link. We'll have the Professor Chris Barnes at University of College London, another professor from London, and also Henry Squire at University of California, Berkeley. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us. I will promote you to panelists who can speak and show your handsome and pretty faces if you wish. And thank you again. I stopped recording. Just 1 second.